Hey guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about forensic biotechnology. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. Forensic biotechnology is a fascinating field that uses the principles of biotechnology to aid in criminal investigations and the administration of justice. So basically, forensic science applies scientific methods to investigate crimes. Why? To determine what happened, identify suspects, and assist in legal proceedings. And it uses different tools and principles of biotechnology. Okay, so it uses following techniques such as DNA profiling. DNA profiling is also known as DNA typing or DNA fingerprinting. In this technique, first the DNA is isolated from the sample such as blood stains, semen or hair, and then it is subjected to southern blotting technique and DNA hybridization with the help of specific DNA probes. So we all know that in southern blotting technique, the DNA hybridization takes place by using specific DNA probes, and the probes correspond to mini satellites in DNA. So DNA fingerprinting is basically a powerful tool and it can be used to help narrow down a list of suspects based on how well their DNA matches the samples that were found at the crime scene. The next tool is PCR. So the PCR is used to amplify small segments of DNA, making it easier to analyze. Since sometimes from the crime spot we can collect very small amounts of sample. So that time PCR helps us tremendously to amplify the sample, to amplify the DNA. Next is gel electrophoresis. So gel electrophoresis separates and analyzes DNA based on their size, and it is used to analyze the DNA sequence. So this is very important to sequence the DNA that we have collected from the crime scene. For that, the gel electrophoresis is important. Next is genetic markers. So genetic markers such as STRs are short tandem repeats. So these are used to differentiate between individuals based on their genetic makeup. Okay, so we can compare these STRs in different samples. Next is biochemical analysis. So biochemical methods such as ELISAs, enzyme-linked immunosorbent assays. So these are sometimes used to detect substances like drugs or toxins in biological samples such as blood or urine. So sometimes we need to find out whether the victim was provided any kind of drugs or toxins, so that we can do by using their blood or urine sample. Next is bioinformatics. So bioinformatics involves the use of software tools and algorithms to analyze biological data, particularly DNA sequences. So once we need to compare different DNA sequences from different samples, biotechnological tools, bioinformatical tools help us. So next is the significance of forensic biotechnology. So why we do forensic biotechnology, how they help us. So first is accurate identification. So by analyzing DNA and other biological samples, forensic biotechnology can accurately identify individuals involved in a crime, providing strong evidence that can lead to convictions. So this is very important and strong evidence because you know that DNA fingerprinting is one of them, since the DNA sequence is identical. Now, next is solving cold cases. So, advanced biotechnological techniques can re-examine old evidence from unsolved cases, leading to breakthroughs years after the crime occurred. Next is preventing miscarriages of justice. So, DNA evidence can help prevent wrongful convictions by proving someone's innocence. So, it helps to prove someone who is innocent and who is not. Next is supporting paternity testing. So, forensic biotechnology is used in civil cases to establish paternity, who is the father, who is the mother, which can be essential for child support and custody decisions. Next is wildlife forensics. So, it helps in tracking illegal poaching and trafficking of endangered species by analyzing their genetic material. So this is all about today's lecture. I hope you liked the lecture. Thank you for watching my video.